Hi everyone, welcome. It is a great pleasure that I can introduce you to Guido Meijer from the International Brain Laboratory. Guido is a researcher trying to understand the brain circuits for complex behavior as decision making. And today he will talk to us a little bit about the challenges that he and the team of scientists that work at the International Brain Lab are trying to solve to understand the brain a little bit more. This is one of the activities planned to commemorate the 50th anniversary of the psychology career of the Bolivian Catholic University, San Pablo. And if you have any questions during the conference, please feel free to send them through the chat. We will take note and do it to our speaker at the Q&A segment. So without further ado, I'm going to leave you with our speaker. Thanks, Guido.
Thank you, Manuel, for that kind introduction. Let me share my screen. There we go. So, hi. Thank you for the introduction and thank you for organizing. Thank all the organizers and Manuel for inviting me here today to give a talk to you. I am a postdoc in the International Brain Laboratory. I'm Guido Meijer, and today I will talk to you about who we are, what our goals are, and what we have so far achieved already in understanding brain-wide circuits for decision making. So the International Brain Laboratory is a global collaboration of 22 different labs, and I myself am situated in the lab of Zach Menen in Lisbon, Portugal, at the Champalimaud Center for the Unknown. Okay, so let's dive right in. One of the reasons why the International Brain Laboratory was founded was because we identified a certain issue in the field of systems neuroscience in particular. And this issue is that a lot of results were not being reproduced. So reproducibility is one of the central tenets of the scientific method. Basically, if you do an experiment with a certain set of methods, and then somebody else, somewhere else, does the same experiment with the same methods, then you should come to the same conclusion, if indeed the underlying thing that you were investigating is true. However, in the field of systems neuroscience in particular, we found that this was often not happening, either because maybe the variability in the results was just so large that when two people did the same experiment, they did not arrive to the same conclusion, but that might just have been because there's just a lot of variability and maybe the sample sizes were not large enough. And then it's still not possible to deduct whether actually you made a mistake and the result turned out to be not true, or it was caused by variability. And also in systems neuroscience in particular, the experiments tend to be very sophisticated and very tailored to the question and very difficult to perform. So it's not being done as maybe in other fields like psychology, where it's people use more standardized assays that have been vetted and a lot of people are using. In systems neuroscience, people tend to perform very specialized experiments, and this makes reproducing them difficult and often does not happen at all. So to tackle this problem, the International Brain Lab was founded, and we are a collaboration of 22 laboratories across Europe, the UK, and the US. And these are all the principal investigators that are part of the collaboration at the moment. We have 22, and 12 of them are experimental, and the other are theoretical. So the underlying idea being that the collaboration actually bridges the gap between experimentalists and theorists, and that theoreticians have a say in which experiments are being conducted at a very early stage, because there is a better open dialogue between these two parts. So our goals in this collaboration is to ultimately to understand brain-wide circuits during a single complex behavior. So behavior has many different aspects and can be seen from many different angles. We have opted to go for a single behavior that will be performed by mice in all the experimental labs that are part of the collaboration. And by doing exactly the same behavior in the same experimental setup with the same protocols, we can then hopefully in the end pool all the data together that is gathered by all the different labs in the collaboration, and thereby gathering a really large data set that otherwise is simply not possible to gather with a single laboratory. And this behavior that we're investigating, I will go into detail about the behavioral task that we're using, needs to be, or is a rich task and a complex behavior where mice are they're exposed to stimuli. They have to learn this. These stimuli are in a certain context with several priors, and then they have to perform actions. And just by nature of their behavior, there will be in different behavioral states, and all these things together constitute our behavioral paradigm. And the way in which we want to investigate how this particular decision-making part of the decision-making field is done in the brain is by, like I said, by standardizing all our experimental methods. So in the end, after we've established this behavior, we are now performing neuropixel recordings. What neuropixels are, I will also get to, which are, they're basically high density electrophysiology measurement probes. And these probes allow us to sample neural activity while the animal is performing this task. And because of this standardization, again, we hope to be able to pool all these neurophysiological recordings together and creating one big data set that is being developed by all the different independent labs. And to be able to all analyze this data, we use common data architecture, so that when the recording is done in the United States, for example, and one is done in Europe, but they both use the same data architecture, which means that anybody can analyze these data, even though they were coming from very different labs. This also allows us to build analysis pipelines on these data that go from the raw data to the processed data in a standardized and methodological way. And in the end, after we have gathered this big data set, we will publicly make it available for anybody to download and to analyze them for themselves. Okay, so let's jump in and talk about the behavior that we're doing. So, like I said, we have picked one single task that all the labs in the collaboration are performing, all the mice in all these labs. And it is what we call a steering wheel task. So basically, there is a mouse, as you see here, and this mouse is holding a wheel. And the wheel can turn left or right, and the mouse is looking at a screen, which is in front of the mouse. Then a stimulus will appear on either the left or the right-hand side of the screen. And the movement of the wheel is coupled to the movement of the stimulus on the screen, whereby if the mouse turns the wheel to the left, the stimulus will move with the movement of the wheel. So it has direct closed loop control on the position of the stimulus. And the goal of this task, or the goal of the mouse, is to position the stimulus into the center of the screen. So if the stimulus was on the right-hand side of the screen, the mouse has to move the wheel to the left, moving the stimulus to the middle, and then receiving a reward in the form of sugar water. Then if the mouse turns the wheel the wrong way, so to the right in this case, the stimulus will move outside of the screen, and there will be some white noise, and the mouse will not get a reward, but instead will have to wait for a certain timeout until the next trial starts. So to do this in all these different labs, we have developed a standardized training rig, which you can see here in this animation. The rig consists of Thorlab components and 3D printed parts And the idea is that anybody who wants to can make a rig like this themselves as well. So all the build instructions are on the website here. So you can order all the parts, you can print all the 3D printed components, and then we have provided instructions for you to put it all together. So if you would feel so inclined, you can just buy these things, print the components, and build one of these standardized rigs yourself as well. So the hardware in this case is also open source. Okay, let's go back to the task that these mice are doing here. So the trick is that if the stimulus were to be very visible, like strongly visible every time, then of course it becomes quite easy for the mouse to do this task. So what we do is we vary the contrast of this stimulus, making it fainter and fainter, and in some cases even completely invisible. So there is a tone when the stimulus is presented, a beep, which indicates to the mouse that the mouse has to make a choice. But in some cases, there will not be a stimulus, or there will be a very weak stimulus. So in those cases, the mouse will have to guess. It knows it has to make a choice because it hears the tone. So we can plot the behavior of these animals in a psychometric curve, as we call this. So bear with me here. On the y-axis, we have plotted the percentage of times the animal chooses right. And then on the x-axis is the contrast of the stimulus. And to the left of 0 here, we have a contrast that is stronger on the left-hand side of the screen. And on the right-hand here, we have when the contrast is higher on the right-hand side of the screen. So when the contrast is 100%, so which is very visible, then in an ideal case, or a good mouse will then also 100% of the time choose right, because that's where the stimulus is. And vice versa, here on the left, when the stimulus contrast is 100% on the left, the mouse will never choose right, so always left. Then here in the middle, there's no stimulus, so the mouse, in an ideal case, will guess 50-50, left or right. And this is how we construct these psychometric curves. And we can distill a couple of metrics from this, like the threshold, which is the slope of this curve, the lapse rate, which is when this is not 100%, that's the lapse rate, and the bias, which is a shift of this curve either to the left or the right. Then we impose another trick at the end when mice have reached this stage of training. And this is, we change the prior probability that the stimulus will appear on the left or on the right. So in blocks of trials, there will be 80% chance that the stimulus will appear on the right. And in another block of trials, it will be 80% chance that the stimulus appears on the left. So when the stimulus is 100%, so at the edges here, we still expect the mouse to see the stimulus and correctly make a decision. But the interesting case here is when there is no stimulus. So imagine that the stimuli are coming from the right 80% of the time. Then, the beep goes, the trial has started, but there's no stimulus. In this case, the mouse is asked to guess, but it will guess more often to the right because it knows that the world is in a state where stimuli are coming more often from the right. And this is what we call a prior. Anybody talking? So the prior is the prior probability that the stimulus is coming from a certain side, but also it can be in the brain because the animal is actually adjusting its behavior and probably also its neural coding to this prior. So this is a trick that we use to investigate what the neural correlates of this prior are in the brain. So this paradigm has been successfully established now in 7 different laboratories. So all these labs have built these standardized rigs and have trained mice in this task using the same methods and the same protocols. And here you can see all the different labs. Every lab has a plot. And all the gray lines are all the individual mice. So this is basically the psychometric curves, as I explained before, of all the different mice in all these different labs. And as you can see, they all look very similar. And indeed, when we do some analysis, we try to decode from which lab a certain animal is from based on its behavior. We cannot do that. And that basically means that the behavior of all these animals is reproducible, as in that we cannot predict from which lab an animal is from based on its behavior in this task. And this allows us to then indeed pool all these data together as if it were one big experiment. And this is recently published in eLife. You can see here the reference below. The data is also open source. So if you are interested in these kind of data, you can go to the paper, download the data, and analyze it yourself. So after we've established that the behavior of the animals is reproducible across laboratories, we aim to go to the next step. And this next step is to actually perform neural recordings in this task, in all the different labs that we have in the collaboration. Our aim here is to record the entire brain. So here on the left, we see the brain of a mouse as seen from the top. Here on the right is the front, and on the left is the back of the mouse brain. And every white dot is a planned insertion of an electrophysiological probe. I'll come to the probes in a second. So these probes are being inserted into the brain at all these different points. And then in the end, spanning the entire left hemisphere. So we only record one of the hemispheres, not both of them, because we assume that they are similar. And the right part of the cerebellum, because the right part of the cerebellum, actually these projections cross over to the left side of the rest of the brain, basically. So these are all the planned sites where we want to record, and we are currently in the process of doing this. We're around 75% completed now at the moment. So this is the first stage of the pipeline, is to plan the recording. Okay, so we say, okay, today I'm going to record this one. So this is the planning. Then you go and you actually do the recording. So here are 2 neuropixel probes that are being inserted in the brain, and we measure neuroactivity based on the planning, based on our planned recording site. Then we have to do histology, because we need to post hoc see where in the end the probes ended up. So you can plan a probe to go to a certain part of the brain, but of course there is this is not going to be 100% accurate, because you have to do that based on skull landmarks, and there's a lot of variation there. So you need to know at the end where you were in the brain, and we do this by slicing the brain, looking at the histology, because these probes are coated with a fluorescent dye, as you can see here. And then at the end, you can go back and see, based on histology, how far off you were to your intended recording site. So that's the sort of loop that we do in recording our brain-wide map, as we call it. So let's talk about the first step here, which is the planning. So we call this the brain-wide map, and the brain-wide map is our intended recording sites all throughout the brain of the mouse. And here I've shown you 2 examples. That here we're looking at the top of the brain. So we see where the probe actually enters the brain at the top. But these probes are angled at a certain angle to cover a certain part of the brain. And we have 2 different angles. One of them is like angling inwards towards the midline, and the other one, and these are superficial. And the other angle is actually angling away from the midline, here, and these are actually deep. So by alternating superficial and deep recordings, we at the end sample neurons from every part of the brain. You can also see that there are some recording sites here on the other hemisphere, but as you can see here, they actually cross over because they have a shallow angle, they actually cross over into the left hemisphere again. So this way, we hope to densely, or at least densely enough, sample from every part of the brain while animals perform this task to get an accurate picture of what the entire brain is doing during this decision-making task. So let me explain a bit about the probes that we use, because not everybody is maybe familiar with them. The neuropixel probes are high-density silicon probes, and they have 1,000 recording channels on the probe surface, which you can see here. And from these 1,000 recording sites, you can select 380 to actually record from. So you cannot record from all of them at the same time, unfortunately, but still 380 recording sites is far more than any probe that's been developed as of late. This probe is quite big, so it's 70 microns wide and yeah, pretty long. It ends in a point and it is like inserted through the dura into the brain, and all these recording sites here then record extra neurophysiological signals, so to say. Here we just see some examples of some features that you can extract from these neurophysiological signals along the probe. And yeah, so this thing is outside of the brain, only this goes in. And the recordings that we do are acute, meaning that we insert the probe into the brain, we record neuroactivity, and then we take the probe out again. And the next day we insert another probe going to a different location. So they are not chronically implanted. We do, this is called acute recordings. So this is the electrophysiological rig that we use. Again, it is standardized, so all the components are standardized, and we have built 10 of these rigs in different labs around the world, and they all look exactly the same. They, so they've been designed so that every part is the same and is positioned at the same place in every different lab. And then we hope that indeed the neural data that comes out of all these different rigs around the world actually is reproducible, or at least generates reproducible results. Here are the two micromanipulators that we use. So we insert 2 probes at the same time. I forgot to mention this. Just to increase the throughput, we insert 2 probes at the same time and record from 2. So this is the just a blow up of what you saw before. So basically this is the head stage. This is this part, the base of the probe, which you see here. And this is the holder that holds it. And then here underneath, you see this long, sort of wire. Like the probe itself is 70 microns wide, which is around the thickness of a human hair. So it's very thin. And 2 of these probes here now are positioned close together. Here we see the skull of the mouse and a small craniotomy where the probe's actually entering into the brain and allowing us to record neuroactivity, which you see an example of here on the right. We have, these are just a small subset of the channels that we record from. So imagine we have 2 times 380 channels, and every channel is a trace here. So this is just a small part of it. And here we see, this is a particularly nice recording. We see beautiful spiking going on, different neurons at different depths here in the brain. So then going to the third step that I explained before, the histology. So our histology is done at Sainsbury Wellcome Center in London by Stephen West, our histology tech. He's really good. Basically how it works is you do several recordings over several days, and then once you're done with recording, you perform a perfusion and you extract the brain from the mouse. And this brain is then sliced. Well, the brain is fixed first in formaldehyde, and then I actually sent the brains to London, and all the histology is done by one person in London who does all the histology for everybody in the collaboration, so that's also standardized. And what Stephen does is he puts the brain under a serial sectioning to photon microscope, which automatically sort of like slides off different slices of the brain while performing 2-photon microscopy. And then this allows us to reconstruct a 3D volume, basically, because we have all the different slices of the brain at different depths, and then you can reconstruct those again into a 3D volume. So every brain that is imaged is registered to the Allen Brain Atlas, which is a standardized atlas of brain regions using some image registration. And then the probes, the probe tracts, are labeled. So here you see in red where a neuropixel probe was in the brain. Again, this is because we have fluorescently labeled the tip of these probes so that they create a fluorescent track everywhere that they go into the brain, basically. So we track these probes using some software, and then the neural activity of these probes is aligned to the histology by looking at neurophysiological landmarks. So finding the XYZ positions of these probes in this track is pretty good, but imagine that you put a probe into the brain and then you retract it a little bit. You cannot see that in the histology, basically. So you cannot accurately assess the depth of the probe. So that's why we look at the electrophysiological signals and the histology, and then align them to one another. So imagine that there is a part of the probe that is silent, and we know that that part of the probe is probably corresponding to a part of gray matter, or white matter, sorry, for example, or some other ventricle, that kind of stuff. And after this alignment is done, we know which part of the probe was in which brain region, and we can then perform like subsequent analyses. So this is just an example of a couple of these reconstructed probe tracks in the brain of the mouse. So every probe track has as you can see, it has different angles. It's at different parts of the brain. And we can accurately then map this to the brain of the mouse, because we have registered it to the Allen Brain Atlas, which is this standardized atlas of the mouse brain. And we can then plot the activity of, or the tracks of these probes into this space, because we know exactly where it ended up. And this is work that's spearheaded by Mayo Faulkner and Oliver Winter, who are staff scientists at the International Brain Lab. So to conclude this part, we have implemented a behavioral paradigm in 7 different laboratories successfully, as is indicated that we could not predict which lab a mouse came from based on its behavior. And we have set up this standardized pipeline for electrophysiology and histology, and basically closed the loop between planning a recording, performing histology, and then looping back to the planning, see how far off we were. All these things are done using standardized analytical and also physical pipelines, as you saw in the histology, in the case of the histology. So then we can start to ask some actual questions. And one of the questions that I wanted to focus on today, because we were focusing on reproducibility, is this one. So when we looked at the behavior, we concluded that the behavior of the mice was reproducible, meaning that it didn't matter in which lab the mouse was, whether it was in the UK or in Portugal, whatever, they all performed the task in the same way. Now you can ask a similar question about the electrophysiology, the neuronal data, basically. So if I put a a neuropixel probe into the brain of a mouse at a certain location in the brain, and somebody else also does this in the United States, then the question is, will we both find the same results? Ideally, of course, the answer is yes, but there are a lot of factors that go into this question and a lot of reasons why maybe this is not the case. So to be able to, for us to create this big data set that is pooled from data across 10 different labs, 11 different labs, we need to first figure out whether our results are actually reproducible. So to this end, we have the Reproducible Electrophysiology Task Force, of which I am a part. It's led by Anne Churchland, Nick Steinmetz, and Wim Paninsky. And we have these other amazing researchers as well in our group. And we set out to answer this question. And we did that in the following way. So every mouse will have recordings from different parts of the brain, depending on what the experimenter did. But the experimenter is required to always make one recording at exactly the same place. And this is what we call the repeated site. So the repeated site here is indicated in red. So this is the brain of the mouse again. So imagine there are 3 mice here with recordings in different places around the brain. Those are all the black tracks. And every mouse will have one of them that will be in a very particular spot. And that's the red one. So it's this one here, this one here, this one here. So by telling the experimenters that they always had to record at least one day at this particular spot, this allowed us to create a data set that that can help us assess whether our results are reproducible over labs. Because of course you cannot accurately compare 2 recordings that are done in completely different parts of the brain, because they're in different parts of the brain, so their results are going to be different by definition. So this is the repeated site. Here we're looking at a cross section of the brain. So this is the left hemisphere and this is the right. And the repeated site is going through posterior parietal cortex here, then going through hippocampus, CA1 and dentate gyrus. And then the bottom part of the probe here is in the thalamus, 2 thalamic nuclei, in particular, the nucleus LP and PO. So this is the planned trajectory of the repeated site probes. However, in practice, of course, they don't always end up exactly at this place, because there is variability in targeting of the probes. There's also variability in skull landmarks, as I've alluded to before. So basically what we do is we insert probes into the brain based on skull landmarks. One of these landmarks is brecma, for example. It's a part where 3 skull sutures meet. And we know that, for example, here the repeated site is 2 millimeters posterior of brecma and 2.24 millimeters away from the midline. And we can punch those coordinates in our micromanipulator and put the probe into the brain, but that is assuming that brecma was indeed at the right location. So there are a couple of issues that create variability. One is that some experimenters might estimate brecma. It's sometimes hard to see where exactly brecma is. It's not super clear cut. And also just there might be variability in the shape and size of the mouse brains. So some brains might be a bit larger or smaller. So that means that the skull landmark relative to the brain is not always exactly the same. And this creates variability. So here we see all the repeated site trajectories as they ended up in the brain. As you can see, there's a bit of a spread around our intended recording site. So this is going to induce variability in our results. So we looked at a bunch of these recordings from different labs, and we tried to see whether indeed the neurophysiological signatures that we saw in each of these recordings is reproducible across them. So one way of looking at this is to look at the LFP power. So as you might know, LFP power is quite high in the hippocampal formation, and particularly in the dentate gyrus. So we have high LFP power in dentate gyrus, which shows as a yellow band here in this plot. So what we see here is every column that we see here, every one of these columns is a neuropixel probe recording. So this is a recording, this is another one, etc. And they have been aligned to the boundary between the dentate gyrus and the thalamus. Now, some recordings were accidentally deeper than intended, like this one, or more superficial, like this one. But by aligning them to brain region boundaries, we could still get them at the same depth relative to one another. And here on the top, you see all the different numbers, and they indicate different laboratories. So these 6 recordings here are from one lab, and these 4 are from another lab, these 6 are from another lab, etc. So here we see data from 5 different laboratories across different countries. And here we see the data of these recordings. So, well, what you'll notice, the first thing you'll notice is that, well, except for maybe this odd case here, all recordings show the same signature. So they all show a band of elevated LFP power at the same depth, so to say. So here we see a band of high LFP power at the same depth, which is corresponding to the dentate gyrus. So this is good news. This shows that we can actually reliably measure LFP power, which is reproducible across laboratories. And we actually also use this LFP power to align the recordings because, as I explained before, we do this alignment of electrophysiological signatures to histology. And one of the features we use is LFP power. So we see that in this case, this alignment was successful and all the recordings nicely lined up. Then there are also other things we can look at besides LFP power. So here I've plotted the same recordings of the same labs, but now I'm showing you the neurons. So every dot here is a neuron, and the color of the dot indicates the firing rate of this neuron. So if the dot is dark in color, and the firing rate is low, and when it's light in color or yellow, it means that the firing rate is high. And the position along the x-axis here, you see that somewhere a bit higher than others, indicates the spike amplitude. So the size of the spikes of this neuron. Then here, these colored squares are the target regions from the repeated site. So basically these regions, so posterior parietal cortex, hippocampus, and the thalamus, using the same color scheme. So here we see the posterior parietal cortex. Here's the hippocampus, the top part is CA1, then we have the dentate gyrus. And here at the bottom, we have the thalamus, 2 thalamic nuclei, LP and PO. And if there's no such square, that means that the recording was actually mistargeted, and it actually missed this target region. It was inserted it ended up in the wrong place of the brain, basically. So we can already see that in general, the thalamus has many more neurons than, for example, the dentate gyrus or the hippocampus, and also their firing rates tend to be higher. So here, for example, we see a lot of neurons with high firing rates. This is consistent with what we know, that in general, the thalamus has higher firing rates, and so we can detect more neurons. And here in cortex, we also see a fair amount of neurons, but as you can already see, not always. For example, here we only pick up 1. So this might be because of other factors. There might be some superficial damage, for example. Some superficial damage during the insertion of the probe, resulting in lower neuronal yield at the cortical level. So how are we then assessing whether these neurophysiological signatures are indeed reproducible over laboratories? So one way of doing that is using the analysis that I will explain to you now. So we can take a certain metric. In this example, I'm looking at spike amplitude in a certain region, in this case, the LP region of thalamus. So we can plot the spike amplitude in microvolts here for all the different labs. So here we have Berkeley, this is us, Champalimaud, we have Cold Spring Harbor, the Churchill lab there, New York University, and Sainsbury Wellcome Center in London. And we can plot the averages of each lab, which is indicated by the short black lines here. And then we can also calculate a overlap average. So it's the average of the lab averages, and this is the red line. So if there is no consistent variation between laboratories, then you expect that the means of the within labs, so the means within a certain lab of the recordings of from the same lab, are very close to the overall mean over all the different labs, meaning that basically it isn't the case that all the recordings from a particular lab are much higher or much lower than the other recordings from the other labs. And you can see in this case that that is sort of true. It's the case. But we see in other features, so for example, the LFP power. So here I plotted the spike amplitude, microvolts here at the y-axis. Here the y-axis now is LFP power in decibels. And you can already see that in this example, there is a certain within lab consistency, resulting in that, for example, the recordings from Berkeley have lower LFP power than the ones at here in Lisbon. So now the lab means, the within lab means, actually are quite far removed from the overall mean. So this would be an example of an electrophysiological feature that is not reproducible, because in every lab there is a different result, basically. And how we quantify this is by using a permutation shuffling procedure, where we calculate the distance between the within lab means to the overall lab mean. So basically this distance, this one, and this distance, and this, which results in a certain metric. Then we shuffle the lab labels. So the colored dots basically are randomly shuffled throughout the different labs. Then we calculate this distance again. And this shuffling is being done like 100,000 times, whatever, a large amount of times. And then we see whether the distance, like the lab to overall mean distance that we found, the actual one, is larger than one would expect based on chance from this shuffled null distribution, basically. And if it is, then that means that this feature was significantly not reproducible in the case that the lab means were significantly deviating from the overall mean, indicating not a good case for reproducibility. So here is an overview plot of all these, all these different neuronal metrics that we looked at, and all the different regions. So the color here indicates the p-value of this permutation analysis that we've done here. So a lower p-value means that the features looked more like this. So they were not reproducible. Whereas a non-significant p-value indicates a situation that's more akin to this, where there was actually no lab-to-lab consistency. So reproducible and not really reproducible. So we have done this analysis for all the different regions of the repeated site separately. Posterior parietal cortex, the hippocampus, C1 dense gyrus, and the two thalamic nuclei. And for all these different electrophysiological markers, so one of them is a neuronal yield, which just means how many neurons that we record in this brain region. Then we have the median firing rate of these neurons, the LFP power, as you've seen here, the AP band RMS, which is basically the sort of like the strength of the high frequency component of the neurophysiological signal. The AP band stands for action potential band. So high frequency components, this is relatively low frequency, LFP power, and the spike amplitude, which is the amplitude of the spikes of the neurons that we find in these regions. And then when we look at the results, is that almost all of them are actually non-significant, except for this one here, which is the example that I've showed here, the LFP power in LP, so LP power is this one. So this one turned out to be significant, but actually all the other combinations of neurophysiological markers and regions were non-significant. So in the end, yeah, at the moment we're concluding that almost all features are reproducible for all the different brain regions that we've looked at, meaning that we cannot predict which lab a recording was done in based on these electrophysiological signatures. So that's what I wanted to talk to you about today. And to conclude, we can say that the standardization of methods in our experiment has resulted in reproducible behavior across different laboratories, as in the behavior of the animal was the same regardless of which lab they were trained in, and a reproducible electrophysiology, meaning that also if you put a probe in the same place in the brain while the animal is performing this task, the results that you get out are indeed reproducible, meaning that you cannot predict in which lab a recording was made based on the neurophysiological signatures. So our future perspectives are to, well, finish the brain-wide map. So as I said, we are around like 75% done with our recording plan, with like recording, yeah, basically the entire brain, hitting all these intended target sites. Some sites are easier to hit than others, as you can imagine. So we're basically now trying to finish up the last parts of the brain-wide map. And those are mostly the parts that are technically the most challenging to record from. So that's what our focus will be right now. This is what we've got the first pass map, and we're also going to do a second pass map where we will assess if there are any parts of the brain that we actually missed in the first brain-wide map. And then we will specifically target those parts of the brain in a second pass, which will be done after the first pass has been completed. So then what we also want to do is develop tools and quality control metrics for the community. So this repeated site data set that we have, we think is invaluable to create quality control metrics, because we have sampled how a lot of different labs perform recordings and how the data from all these different labs looks like. So we can basically accurately say, okay, these are the quality control metrics that a recording has to abide by to be able to actually provide meaningful results. And this is much harder to do if you are just a single lab doing recordings, because then you have no estimate of lab-to-lab variability between labs, basically. But because we have this large broad data set, we can actually do that. And we will release these tools and these quality control metrics open source for the community, so that basically everybody can also use them and yeah, make use of the data that we've collected. And of course, all the data will be open source and publicly available. So after we finish the brainwave map and the reproducible EFIS, we will publicly make all the data available so that everybody can download the data and actually analyze it themselves if they want to. Okay, well, that was shorter than I expected. That was it for me. I wanted to thank everybody in the IBL and thank again, Manuel, for inviting me here. Here are all the PIs in our investigation, in our collaboration. We have a lot of researchers, PhD students and postdocs. We have an amazing group of staff that help us putting down all the pipelines. We have some external partners, and I'd like to thank our funders, the Wellcome Trust, Science Foundation, and INCF. If you want any more information, visit our website, find us on Twitter, or check out our GitHub for all our open source code. Thank you.
Okay, thank you, Guido. It's been a great talk, very interesting. We have a couple questions from the audience, and we would like to do it right now. First of all, are there less invasive methods? Are the less invasive methods equally reproducible, or do they involve greater variability?
So it depends a bit on what you call less invasive. Putting a probe in the brain is, of course, quite invasive because it goes into the brain. We are now starting a new project with mesoscopes, and mesoscopes are basically 2-photon calcium microscopes that can measure a large part of the cortical surface. And in that case, you do have to implant a cranial window. So in that sense, it's still invasive, but nothing is actually inserted into the brain. It's all done with microscopy. So this is something that's currently ongoing. We have not actually gathered any data. So the short answer is we don't know yet. We might even start some human experiments as well with fMRI. And when we do that, then we'll be able to more accurately answer this question. But at the moment, we just don't have the data yet.
Okay. Can we translate the results of these experiments to other species, like other mammals or humans?
Yeah, so we hope so, of course. I think there also in humans, people do intracranial electrophysiology akin to neuropixel recordings. For example, with people with epilepsy, they get implanted electrodes in their brain. This is harder in reproducibility because these electrodes are placed for medical reasons. So you cannot really target the same place in the human brain every time. So that's a bit of a difficult question to answer. But for monkeys, we definitely think that these results are translatable, and we might even start some monkey recordings as well. And for humans, I think they are, but actually proving that they are might be quite challenging unless you go into fMRI, for example. Then it's a different story.
Sure. Another question is, how is data science and machine learning being used for the research done on the International Brain Lab?
Yeah, so we have, like I mentioned at the beginning, we have theoretical and experimental labs. So the experimental labs are mostly focused on actually doing the recordings, but the theoretical labs have a pretty strong analysis focus, and a lot of them are actually performing machine learning and other data and analytical procedures. Yeah, so as you can imagine, this data set is a goldmine for machine learning because it's so large and it's very diverse. So there are several different ways in which these kind of analyses are being used. One of them is that we decode from neuronal activity certain behavioral parameters. So we can decode from the brain, like what was the choice of the animal, what was the prior probability of the stimulus being left or right. this one is of particular interest to us. So basically we say, given the neuronal activity, can you predict whether the stimulus had a larger chance coming from the left or from the right? And we do this, try to find in the brain where there's a neural correlate of the prior, as we call it. So we think that the brain always has an idea of the state of the world, the state that the world is in. And when this changes, it will update its, basically its internal prediction, its internal model of the world. And so far, it's not known how the brain does this, or even which brain regions are involved in doing this. So that's why we use machine learning to try to pinpoint a part of the brain that is responsible for reflecting the state of the world, basically, for reflecting the prior in this case.
Sure. Another question that is kind of related is, what kind of mathematical skills do you need in order to work with us with all this information?
That's a good question. I don't think that pure math is really necessary. I don't know a lot of math. I'm a biologist. So nowadays, with the advent of Python and all these analyses packages, it's actually not really required that you have to code a lot of math by hand. So if you look at scikit-learn, for example, which is a great Python library for doing machine learning, it makes it really easy to do quite advanced higher dimensional analyses or dimensionality reduction or fitting machine learning models. Manuel can attest to this. He has done this in the Neuromatch Academy. It's made it quite simple and user-friendly. So I think it's more important that you understand the mathematical principles that underlie a certain computation that you do, that you understand the gist of it, you understand what it does at a high level, but you don't really need to know the nitty-gritty implementation. So I would say that pure math is not really necessary. It's you need to have a certain intuition for the analysis techniques that you use.
Awesome, great, great insight. Is any new discovery already been made regarding this decision-making process with the IBL experiment?
So we are still in the process of gathering all the data, and it's still in the early stages. What I can tell is that there's at least not one brain region. So one of the big questions that we set out to answer was where and what is the neuronal correlate of the prior, of the Bayesian prior, basically, in the brain. And well, the only thing that we know for sure that it's not one region that does it, which is almost always the case in these kind of things. You ask, oh, which part of the brain does it? And then you find out it's distributed throughout the brain, and it's not like one, there's not one region that is responsible for actually updating updating our world model, or basically, yeah, how do you say, constructing this model of the world and keeping track of changes in the environment. It doesn't seem to be, we are not able to pin it down to a single region that does this. And it's also not very strong. Like we were expecting to, we threw a bunch of machine learning models at the data, and we were expecting to find a pretty clear neural correlate of this prior. So far, we have not found very strong conclusive evidence for this, indicating that this neural code is probably quite subtle. It's not something that is very evident from a first pass. So yeah, it's not a very satisfying answer, I realize. So I would say no, we have not made any groundbreaking discoveries yet, but we're working on it.
Sure. Are the IBL plan to work with maybe other type of animals, like other type of mammals or other type of experiments, maybe experimental paradigms?
Yeah, so we recently got a U19 grant, and in this grant proposal, there is also indeed the plans to go for other species, in this case, monkeys. To be honest, I don't know the details of like which labs are going to do this, but yes, we are planning to expand to monkeys, and we got the money to do this quite recently, so the plans are not yet completely finalized. So I think at the moment humans are not yet on the map, on the roadmap, but it is completely possible that in the future we will also perform maybe EEG, fMRI, MEG, one of these methods, and actually do the same, a similar thing in humans. Yeah.
Sure. Does the IBL work with the simulation methods like optogenetics?
So I've not touched upon this, but basically there are two parts of the IBL. One is the collaboration-wide goals, which is the first pass map, like recording the entire brain, that kind of stuff. Then besides that, individual researchers in the IBL can have personal projects. And in their personal project, a researcher can do what they want, basically, but still within the IBL framework. So they can use the pipelines and that kind of stuff that's being developed by IBL on the rig, but then to answer a more specific question that they are personally interested in. And in those personal projects, this is happening. So in my personal project, I'm interested in serotonin. So what I do is I take the task, the IBL task, then I inject a virus in the dorsal raphe nucleus, a generodopsin virus, I implant a fiber, and then I actually stimulate serotonin release while animals are performing this task. And like my hypothesis is that serotonin is actually influencing the way in which mice use their prior beliefs, and how quickly they update their prior is, I think, influenced by serotonin in this case. So yeah, in all these personal projects, and I know for several others as well, people are doing optogenetics. Yeah.
Awesome. There's another one. How long does the animal surgery take? How long can these recording probes last in the mouse, in the brain of the mouse?
So the surgery is done basically the day before. So we make a craniotomy in the skull, and then well, the brain is then exposed, and the brain is then covered with a certain protective coating, which is called Duragel, which protects the brain from the outside world. Then the next day, sorry, those are my cats. Then the next day, the craniotomy is there, but we don't have to do another surgery because the craniotomy is already made. Then we insert the probe into the brain and do the recording. The recording lasts around an hour and a half, I would say. And then when the recording is done, I basically pull the probe out of the brain again. And this Duragel, this protective coating, is sort of like closing. When the probe is out, it closes and protects the brain. And then the day after, I can come back and put the probe from a different angle, for example, in the same craniotomy. And this can go on for like maybe 3 days, 4 days at most.
Okay. So right now the NeuroPixel probes are inserted manually, right? You inserted.
Oh, well, it's a robot arm, but yeah, manually. Yeah. Like I control it with like with some a remote control, basically.
Okay, yeah, I was just about to ask you that because maybe a robotic approach, if you guys were considering like a robotic approach for the task, like, I don't know if you've seen the presentation that Neuralink did like 2 years ago, something like that, with a robot.
Yeah, so that we don't do. Like the surgical robot that Neuralink has is very advanced. And so in that sense, we still do it manual, even though we do have a robotic micromanipulator. It's still like the craniotomy is done by hand. And then basically I look into the microscope and I control the robot arm and put the probe in. So it's semi-manual, I think. Although there are plans to buy also a robotic surgery device that can make the craniotomy in the skull, but then a robot does it. So the robot drills into the skull, it's starting to look a bit like what Neuralink is doing, but not exactly. It drills out the craniotomy in the skull so that you don't have to do that by hand. So the chances of making damage are much lower when it's done by a robot like this.
Sure. There's a question that says, why is open source important for the International Brain Lab in Neuroscience?
So it's that's a good question. So we believe that open source is important for the scientific process in general. So we think that, it's basically just one of the principles upon which the IBL is founded. The idea being that knowledge should always be open and available to everybody, because we want to steer neuroscience away from the situation in which every lab basically does their own experiments in a closed environment that is not being shared with the world, because then also it becomes quite hard to check whether what they do is actually correct, and like people reinvent the wheel every time. If you don't share hardware and software, then every time that somebody wants to do a similar experiment, they have to themselves invent things that have been invented before already. So we believe that you should always make hardware open source and available to everybody, so that it just is a much more efficient way of doing science on a global scale, basically. Because now if somebody wants to do a task where a mouse moves a wheel, they don't have to design a rig or write task code, whatever. They just copy what we do, because we have provided them with all the necessary materials and the code to run the task and that kind of stuff, which we think is the way that science ideally should be done. And we realize that a lot of people are not doing this open approach, but hopefully when this is becoming more normal, then more will be shared, and then the whole process will just be more efficient, basically, and better, ideally.
More accessible.
Yes.
So this question is related to that. Is there a way for new labs in LATAM or Bolivia to join the IBL experiments in the future?
Yeah, so at the moment, we are actually not expanding labs. So we are sort of now at the moment we're focused on performing this brain-wide map. So we're sort of locked into that pipeline. And it's been decided that at the moment we want to keep it with the amount that we have now. But in the future, there are definitely plans to expand the collaboration and also make it open for other labs to join. There's still some debate on how we're going to do that. It's difficult because if you grow, then also communication channels will become more difficult to maintain. So there's currently a task force that is tasked with basically seeing how the IBL can grow, what steps we need to take to enable this, if we have the resources, that kind of thing. So we first wanted to basically do a bit of a proof of principle. So this brain-wide map that we're doing now is sort of like a proof that the way that we do science sort of works. And after we've established that and done that, then indeed we hope to expand and open it up.
Sure. And what can you recommend to us if we want to pursue a PhD in neuroscience in a related field of the IBL research?
So, well, I think the most important thing is that you gather the necessary knowledge for doing a PhD. So I think things like the Neuromatch Academy are great examples of ways in which you can get in touch with the more, with the neuroscience community, for example, and sort of see also what other labs are working on. Then it's kind of difficult to give one answer to this question because it depends strongly on which subfield you are interested in. So if you want to be more on the experimental side or more on the theoretical side. If you are interested in doing a PhD on the experimental side, then gaining experience in techniques is, I think, very important. So when you start applying for PhD positions and you can show that you have experience with the technique that you're going to use in your PhD, this is going to be a huge plus for you. So imagine you're interested in neuropixel recordings or whatever, then if you can do an internship somewhere in a lab where they do that, and you don't even have to do them yourself, just like do an internship somewhere where they do these things, and then you can already say that you have experience with them, which will help you a lot on the job market. And the same thing with theoretical, I guess, is it also, but you can also be a bit more autodidactic in that case, where you think, oh, I'm interested in machine learning, whatever. Make sure you get experience in what you are applying for. So when they say, oh, do you know about this machine learning technique? You say, oh, I did a summer school or a course, or I did an internship. Anything that you can do to show that you have the relevant experience is going to help you a lot.
Which is kind of contradictory, right? Because you want to do a PhD because you don't know things and want to discover things. And probably you don't have that experiences in labs in Latin America or Bolivia.
Unfortunately, the job market is pretty tense at the moment. So, of course, you're right. Ideally, you don't need experience because you are going into a training trajectory. But yeah, in my experience nowadays, it's quite difficult to find a job because there's just not enough. There's not enough positions for everyone. So then you need to find an edge. And I agree with you. In an ideal world, that it's not necessary, but fortunately, it is.
There's a question. What type of programming languages are used for your research? You mentioned Python, I guess. Maybe other
everything
ones?
in Python.
Everything is in Python.
Yeah.
Great. You mentioned NMA, the Neuromatch Academy. Can you talk a little bit what is NMA for the people that don't have any idea, and maybe tell a little bit about your experience as a mentor and all that?
Yeah, so like I said, I was a mentor for Manuel when he was doing the Neuromatch Academy. And the Neuromatch Academy is a global summer school, completely virtual. It's, I think, correct me if I say something that's wrong, Manuel, you know better than I do. I think it's like 3 weeks or so, where you do an intense course on several topics. It has a focus on computational neuroscience. So you learn a lot about machine learning, deep learning, deep nets, that kind of stuff. And the people who give it are, they're just, really big people in the field. They're very good. And I think it's a great opportunity to sort of network because you see a lot of people that you might otherwise not encounter. And because it's completely virtual, it's also very nice that it reaches a very global and diverse group of people, basically, that maybe otherwise would not be able to all come together at a physical location. Yeah, also because sometimes you just don't have the funding to travel to a conference in America, for example. And in this case, it gives you this opportunity, which is really nice in sort of leveling the playing field for a lot of people. I mean, I would not be giving this talk to you if it had not been for Neuroscience Academy, which is just a nice example, I think, of how such a virtual conference breaks down otherwise physical boundaries and yeah, brings together people that maybe otherwise would not have met, which is nice. And just the content of the course, I think, is really good. Like I said, it has a bit of computational focus, but you really have great teachers there that know their stuff. And also you do a personal project within the summer school where you actually get hands-on experience with some of these data sets. And one of these data sets is NeuroPixel recordings as well. Nick Steinmetz, who is part of the collaboration here, he has gathered a large NeuroPixel data set, which is open source. And in this case with Manuel, he has gone in and analyzed these data, which I think is a great way to get some hands-on experience with data analysis, which data analysis of this kind can be quite complex because of the nature of the data. So I think in that sense, it's very important.
Sure, great. This is one of the last questions. Where could I find more to study about reproducibility and the things that the IBL is doing? Okay.
So you can find a lot of information on our website here. The reproducible EFIS paper is not out yet, so it's very much a work in progress. We will have a poster on SFN. So if you are partaking in maybe the physical or the virtual part, I think it's both this year, of SFN, you can find us there. We will have several posters, and one will be from the reproducible EFIS task force. So you can go to our virtual poster or our physical poster if you want to know more. Otherwise, yeah, you can just do some literature research on the topics of reproducibility. And yeah, if you just go to Google Scholar and type in reproducibility crisis in neuroscience, you'll probably find a lot of hits.
Awesome. I read this in your blog, in your personal blog, that you're interested in psychedelics and hallucinogens for treatment of psychological conditions. Mm-hmm. Can you share with us some of your thoughts regarding that? Do you think it will be a viable treatment in the future, maybe?
Yeah, so the reason that I became interested in psychedelic and psychedelic-assisted therapy is because at least the classical psychedelics, they all act on the 5-HT2A receptor. And this is a serotonergic receptor. So that's how I got into it. I started doing research into serotonin. And then I basically quickly found out that all these psychedelic compounds act predominantly through the serotonergic system. And as you might know, depression is also very much associated with a dysfunction of the serotonergic system, because almost all antidepressants are selective serotonin reuptake inhibitors. The problem is that we found out that depression has something to do with serotonin because SSRIs, or selective serotonin reuptake inhibitors, work. It wasn't the other way around. It's not that we have a very good understanding of how the serotonergic system is involved in generating depressive symptoms and thought, okay, that's why we have to give patients with depression SSRIs. No, they just gave people with depression a bunch of different drugs. Then they realized SSRIs work. They're like, oh, probably depression has something to do with serotonin. But of course, this is the wrong way around. So I'm interested in trying to find out how exactly serotonin is involved in depressive symptoms, and also then trying to learn from psychedelic treatments, for example, how this can be leveraged to then alleviate these depressive symptoms. We know that psychedelic-assisted therapy works because of all the papers from, for example, Robin Carhart-Harris at King's College in London. He recently again published a paper like a couple of months ago, very interesting, where they did a head-to-head comparison between SSRIs and psychedelic-assisted therapy. Basically, what they found is that with people with very strong depression, they worked equally well. So meaning that having 3 sessions of guided psychedelic assisted therapy had the same antidepressant effect as being constantly on SSRIs. Now, SSRIs are not very nice. They have a bunch of side effects. You have to take a lot of them. It's basically like taking a hammer and bashing it on the brain and hoping that something good will come out. It's not great. And psychedelics, on the other hand, have no side effects. You only have to do it a couple of times, and the effects are equal. So when they asked the patients what they preferred, SSRIs or the assisted therapy, psychedelic assisted therapy, all of them said that they preferred the psychedelic assisted therapy, which I think also is a much better way of approaching things like depression, instead of just giving a bunch of very strong drugs that we don't even know why they work. So I think by investigating how psychedelic assisted therapy actually has an antidepressive effect, we can in the future develop better and more selective drugs than SSRIs to treat depression.
Awesome. So you are very optimistic regarding the future of this field.
I am, yeah. I think it's a growing field, because of course, in the 70s there was a large knockdown of the whole field, but it's re-emerging, and I think there's still a lot of stigmas to be broken. But I really do think it's a growing field. Yeah.
Awesome. So this is the last question. It's a little bit more personal. What inspired you to pursue a career as a researcher in neuroscience?
Yeah, that is a good question. So I sort of see neuroscience as one of the largest remaining mysteries, I think, that we as humans face. So there are a couple of areas in science that are just, in my mind, still completely open, or still we don't really know much about it. And I would say that is neuroscience and quantum physics. And quantum physics, I mean, I'm not a physicist. I can barely do math. So I was like, nah, let's not do that. And so then I became really interested in neuroscience. First I did biology, and then I did neuroscience. And what I find so interesting about it is that it's very relatable to everybody. So basically everybody has a conscious experience, right? So not everybody sees quantum entanglement all the time. So quantum physics is a bit further removed from our day-to-day experience to actually to generate this sort of like relatedness to the questions. Whereas everybody is conscious all the time, well, not all the time, is conscious, and can therefore ask themselves these questions like, why am I conscious? How am I conscious? It is, I think this is what makes it such an interesting topic and such an interesting field. Because it's sort of like a shared a shared experience, and also a thing that everybody can ask themselves, which I think is cool.
Awesome. What are your views regarding this type of companies like Synchron, Neuralink, Braingrade 2 for the future of neuroscience?
Yeah, so I think a lot of people don't like Neuralink. I get that. I sort of see, I'm quite impressed with the work that they do. I think the biggest problem is proprietary. The problem is that almost everything that Neuralink develops is closed source. So you do not get access to the hardware they develop, the software they develop. Everything they do is proprietary, which makes sense because they're a company for profit. So of course they're going to be proprietary. The problem is that this will not really translate well in advancement of the field as a whole, because when Elon Musk develops this robot that can insert neuropixels into the brain without making any damage, that's nice, but it doesn't really help me because I don't know how to build such a robot, and I probably never will. So I think these companies are great, and some of the claims have been overblown, like Elon Musk saying that we don't use language in 10 years is of course, that's not true. But I think that they are on track to actually be very important in helping people with paralysis, for example. Like such an implanted chip that they develop has a very good possibility to actually help people that have lost control over their limbs, that kind of stuff. And yeah, because they have a lot of money and a lot of technology, I think they probably will be much better at that than other people who are trying to do the same thing. The problem is that they often claim to be the first in something, whereas they are actually not. So this is a main criticism on Neuralink, where maybe you remember the monkey playing Tetris with their mind. You know, we've been doing that for 15 years. It's not new. So a lot of people don't like that, which I totally get. But on the other hand, they are doing it at scale, and just with a lot more resources. And you can say what you will about that, whether it's good or bad, but I do think that they will be able to help in the end a lot of people with paralysis, which is a good thing, I'd say.
Sure, awesome. Is there a way for people to reach to you if they have following questions or?
Yeah, you can find me on Twitter or go to my website, which is just my name, guidomeijer.com. And there you will find my email address and that kind of stuff.
Sure. Awesome. It's been a pleasure, Guido. If you visit Bolivia again, maybe in the future, please let us know. And we make sure, also make sure if you to visit our university and our lab. If you're welcome whenever you want.
Last time I was in 12 years ago, so I'm due for another visit.
Awesome. We'll be waiting for you. It's been a pleasure talking to you again. Wish you the best. Thanks for everything.
Thank you so much for hosting, organizing, and for inviting me.
Thanks, Guido, and thanks everyone for being here. So
thank you. Bye-bye.
Bye-bye. Bye.